Once again, I want to welcome us back to the class. We are sorry for the break in transmission. As I said earlier, what we want to have this afternoon is the cell and its environment. And the basic points we should all understand here is that we want to see how the cells behave in their environment. Of course, we know that the cells will live either singly as unicellular organisms, or they can also live together as multicellular organisms. They can also live in colony, they can live in filaments. We have discussed that before now. Now, what we are going to be talking about this afternoon is how the cells behave in the environment, and the basic things we must understand here, or the basic terms we should understand are: 1, diffusion; 2, osmosis. Then we can now be talking about other forms of diffusion that we have. I want you to also note that even osmosis is a type of diffusion. Now, what do we mean by diffusion? We are going to take the definition quickly, then we can take some instructional materials, some instructional videos to help us buttress on our point. Now, what is diffusion? Diffusion is defined as a process by which molecules or ions of substances move from the region of high concentration to the region of low concentration. I will take it again. Diffusion is the process by which molecules or ions of substances move from the region of high concentration to the region of low concentration until they are evenly distributed. Now, it means that there are always two concentration of substances. Two concentration of substances, the high concentration and the low concentration. Or basically, you can say the higher concentration and the lower concentration. Now, there are two substances now. Molecules will move from a substance having a high concentration to a substance having a low concentration. You understand me now? Or ions as well. Ions move from region of high concentration to region of low concentration. That is basically what we refer to as diffusion. And of course, we should remember that the movement, it moves because the two concentrations wish or want to become even. That is, they want to have the same concentration. Let's say, for example, we have a substance of water. We have added sugar to that water. Then there's another water that is ordinary. We didn't add anything. So diffusion is going to occur in such a way that the one we have in the substance or the solution of sugar will become the same as the one we have in that ordinary water. That is basically what we mean by diffusion. Now let's now say, for example, you have your perfume in your house, or let's say air freshener. Gaseous air fresheners. You use it inside your room at a particular edge of your room. Before you know it, you will start perceiving the odor at the other edge of your room until it fills the whole place. That's what we call diffusion. You should try to get it, try to understand it. Now, diffusion can occur in any substance. It can be in the solid, in the liquid, or in the gaseous. Diffusion can occur in the solid, in the liquid, or in the gaseous. Now, let's now talk about in terms of cell. In terms of cell, from region of high concentration to the region of low concentration. Let's say a cell, particular cell is put in an environment. Of course, you know that that cell contains some cytoplasm in the cell. And the cytoplasm contains substances. Now, what happens is you put it in that particular environment. The cell will tend to release some substance from inside the cell into the environment. So that before you know it, both the cell and the environment will have the same concentration. Okay? Or if you now put a cell in a particular environment that has a higher concentration, what happens is the substance in that environment tends to move into the cell to make the cell what? To make the cell and the environment the same. That's what we call diffusion. Just basically understand that movement of molecules of or ions of substances from the region of high concentration to the region of low concentration. That is the basics. Now, let's see, what are the factors that affect diffusion? What are those things that contribute to diffusion? Or what are those things that make diffusion faster? That is what we refer to as the factors that affect or control diffusion. Number one, we talk about the state of the matter. The state of the matter. Now, what do we mean by state of matter? Because in your chemistry, you will have been taught state of matter. They will tell you matter exists in three states: the solid state, the liquid state, and the gaseous state. Yes, you are right. Now, understand that when molecules are in the gaseous state, they tend to diffuse faster. That is, gaseous is higher than liquid, is higher than solid. When molecules are in the solid state, their diffusion is very slow. But when molecules are in the liquid state, the diffusion is higher than that of the solid. And when molecules are in the gaseous state, the diffusion is very, very fast. So it means that the state of matter determine or affect the rate of diffusion. Number two, we talk about the molecular size, the size of the molecules. The size of the molecules also affects the diffusion rate. That is, the higher the molecules of that substance, the lower the diffusion rate. And the lower the molecules, the smaller the molecules, the faster or the higher the diffusion rate. Number three, we talk about the concentration, differences in concentration. Let me come again. I said number one, the states of matter. Solid state is slower or is the slowest. Liquid state is fast, while gaseous state is the fastest. Number two, I said we have molecular size. The lower the molecular size, the higher the diffusion rate. And the higher the molecular size, the lower the diffusion rate. That's number two. And number three, I said concentration, differences in concentration. The higher the difference in the concentration, the higher the diffusion rate. That is, from one cell to the environment or from one cell to another cell, okay? If the difference in the concentration is high, as the example I used before, you have salt and water. If as much the salt you have in that salt water compared to the amount or the ordinary water, the diffusion will be faster. Do you understand me now? The diffusion will be faster because the concentration of one is very high compared to the concentration of the other. But it now seems that as if you have two substances having almost the same concentration, the diffusion will be very slow. We get it now. So that is that. Then lastly, temperature. Temperature affects. Temperature is the degree of coldness or hotness of a particular substance or a particular body. So it means that when the environment is hot or warm, the diffusion rate will be very fast. But when the environment is cold, the diffusion rate will be very slow. I don't know if you understand that. Now let's see this example. If you have cold water, take cold water and pour sugar inside it. You will discover that, okay, cold water in a glass cup, pour sugar inside it. You see, it will take you some time before the sugar will dissolve. But if you have hot water, the one that is hot enough to prepare tea, pour sugar inside it. You may not even need to stir before the sugar will melt. You understand me now? So it means the rate of diffusion is affected by temperature. The higher the temperature, the higher the temperature, the higher the diffusion rate. The lower the temperature, the lower the diffusion rate. I think that has been sorted. Now, another form of behavior of cells in the environment is osmosis. Now, osmosis is a form of diffusion, but people normally look at it as if it is the opposite of diffusion. No, it's not the opposite of diffusion. It is a form of diffusion, but the definition makes it look as if it is the opposite of diffusion. Now, what is the definition of osmosis? Osmosis is defined as a process by which substances or molecules of substances move from the region of low concentration to the region of high concentration. The movement of substances or molecules of a solvent from the region of low concentration to the region of high concentration. See, it looks as if it is the direct opposite of diffusion. Okay, diffusion is movement of molecules or ions of substances from the region of high concentration to the region of low concentration. But for osmosis, the movement of substances or solvents or molecules or ions or whatever you want to call it from the region of low concentration to the region of high concentration. Okay, now let's now see what are the conditions necessary for osmosis. Like we have the factors that affect diffusion. We are also going to talk about the factors that affect osmosis. Number one, we talk about the presence of stronger solution. The presence of a stronger solution. For osmosis to occur in a cell or in a substance or in anything at all, there must be a stronger solution. There must be a stronger solution. Okay? Now, like we have it in diffusion, we said differences in concentration. Here it is, we are talking about solutions now. Let me just cite this note of warning quickly. In a cell, the cell is made up of the cell membrane and the cytoplasm. The cytoplasm is the liquid inside the cell in which we have various organelles. The organelles, we have the nucleus, the cytosol, the cytoskeleton, the ribosome, the lysosome, mitochondria, name it. Okay, now that liquid in which all these organelles are floating is called the cytoplasm. Cyto means cells, plasm means liquid or plasma. So the liquid of the cell is the cytoplasm. You understand me now? So that liquid inside that cell wants to move out of the cell, or we say liquid outside the cell wants to move into the cell. That is basically what we refer to as osmosis. Now, when a particular solution is high, it tends to pull in, it tends to draw the solution that has a lower concentration into itself. So movement of solutions or solvents from the region of low concentration to the region of high concentration. I said the presence of a stronger solution must be there. It is a factor or it is a condition that is necessary for osmosis to take place. Number two, we have the presence of a weaker solution. We have the presence of a stronger solution, number one, for osmosis to occur. Number two, we have the presence of a weaker solution. That is, you must have a strong solution and you must also have a corresponding weak solution. Then lastly, there must be the presence of a selectively permeable membrane. Those are the three conditions that must be met before osmosis takes place. Number one, the presence of a strong solution or a stronger solution. Two, the presence of a weaker solution. And three, the presence of a selectively permeable membrane or a selectively permeable surface. That is a surface that will allow molecules to move from one side to the other side. That is what we refer to as selectively permeable. It must be permeable, that is, it must allow liquid to pass, but not all the liquid. It must select the ones that should pass and the ones that should not pass. Now, okay, that is that. Now, cell as an osmometer. There are three things I want us to understand here. There are three things. There are three types of solution we must understand. There are three types of solution we must understand. When we're talking about there must be a weaker solution, there must be a stronger solution. The solutions are of three types, be it weak or strong. Number one, you have the hypertonic solution. Hypertonic solution. What do we mean by hypertonic? Of course, hyper means something that is very high. You would have heard of hypertension, high blood pressure. High blood pressure, hypertension. There's another disease called hypotension. Hypotension means low blood pressure. So when you hear the word hyper means high. So we have hypertonic solution, a solution that contains a relatively large amount of concentrated solution, a substance having high amount of concentrated solution. That's what we call hypertonic solution. Then the opposite of hypertonic solution, we have the hypotonic solution. That one has very low amount of concentration. That is the hypotonic solution. Then lastly, we have the isotonic. Iso means something that is standing at the middle. It's not too high and it is not too low. So because it is at the middle, we call it the isotonic. Tonic, of course, you know tonic. When you say blood tonic, this tonic and that. Tonic simply means liquid or a solution. So hypotonic is a solution that is having a very high concentrated solvent, while hypotonic is a solution that has very low solvent. Then isotonic is in the middle. It's not high, it is not low. That's the normal. You understand that now? So that is that. Now, there are some, there are some experiments there that we should carry out, but unfortunately, I am not in the laboratory now. I will have done it and got the video across to you to make your work easier. But even at that, I can still use my mouth to explain. There is something we call potassium permanganate. Potassium permanganate, that is potassium tetraoxomanganate 7, KMNO4. It's called potassium permanganate. It has a kind of violet color. It is found in the laboratory. So what you do is just get the potassium permanganate, get water, just drop it inside the potassium permanganate. Drop the potassium permanganate into the cup of water or a glass of water or a beaker of water, and what happens, it goes down. Then before you know it, it will start dissolving. And as it is dissolving, it will be filling up the glass of water little by little from the bottom. And before you know it, it will have filled up the whole place. It means the concentration of that permanganate is very high at the bottom. Before you know it, it starts spreading little by little, spreading until it fills the whole liquid. That is an example, that is a way of demonstrating diffusion. You understand me now? Another very simple way is when you use, I've mentioned that before, when you use your room sun, your air freshener, or you use your perfume, or somebody is cooking in the house, you're outside the house, before you know it, you start perceiving the odor, the aroma from the kitchen. So it is diffusion that is occurring there. Now, there is another type of osmosis. There's another type of osmosis which we call the plasmolysis. Plasmolysis. What do we mean by plasmolysis? Plasmolysis is a combination of two words, plasma and lysis or lysis. Plasma is the liquid in the cell, and lysis means to break out. So when we say plasmolysis, it means a cell, the liquid inside the cell moves out of the cell to the point that the cell shrinks and it's being disfigured. So in that type of situation, when a cell is put in an hypertonic solution, when a cell is in an hypertonic solution, that is a solution that has a high concentration, you put a cell inside it. What happens to that cell? The solution inside the cell or the liquid inside the cell will move out. The liquid will tend to move into that one that has a very high concentration. The liquid will move out from inside the cell back into the environment or into the surrounding liquid. And before you know it, the cell will start to shrink. All the water will move out of the cell and the cell will now shrink. That's what we call plasmolysis. Another one is hemolysis. Hemolysis. Hemolysis too is a combination of two words. We have the hem, H-A-E-M, and we have the lysis. Hem means blood. Hem is gotten from the word blood. That is the meaning of blood. Of course, you will have taken your Didion hemoglobin syrup. Hemoglobin is a protein of the blood. So whenever you hear the word hem means blood. So when we say hemolysis, it means a blood cell put in an hypertonic solution, what happens to it? All the liquid in that cell, in that blood cell, tends to move out of the solution. I mean, moves out from inside the cell into the surrounding solution. And before you know it, the cell will what? Will shrink. That's what we refer to as a hemolysis. Okay. Now, lastly, I mean, we have another one, turgidity, or you call it turgidity. Turgidity, Turgidity, is a direct opposite of hemolysis and plasmolysis. Turgidity means when you place a cell into an hypotonic solution, you place the cell in an hypotonic solution, what happens? The solution, the surrounding liquid, will tend to move into the cell because the solution inside the cell is hypertonic. Now, the solution outside the cell has a very low concentration. Therefore, they tend to move towards the cell that has, the part that has a high concentration. So the surrounding cell, the surrounding environment, which contains hypotonic solution, the solution there moves into the cell, and before you know it, the cell will become what? Big, becomes fat, becomes robust, becomes enlarged. And as it is being enlarged, what happens to it? The cell membrane will be stretching, and the water will be coming in. The water will be coming in. The water will be coming in. The water will be coming in, and it's going to reach a particular point. What will now happen? It's going to burst. So when it bursts, that is another type, which we call flaccidity. If after it has bursted, the cell has burst, then it becomes flat again. That is what we call flaccidity. You understand me now? So it means that when the liquid outside, the solution outside is moving into the cell continuously without stopping, the cell becomes turgid, as you want to call it. It becomes robust, becomes large, becomes fat, becomes big. Then after a long time, what happens is going to what? It's going to burst, and it leads to what we call flaccidity. Now, let me quickly share with you a video that will help to buttress what we have been saying since morning. Excuse me, let me get it from here. Excuse me. Still trying to pinpoint it from here. Here. Gallery. No. I'm going to use share video. I'm going to share screen. I'm going to share. Let me see if I can share camera. Okay. So trying to share video. Share